Hello students, today we are going to learn 2 points. One is the mutation in genetic code and second is the structure of a tRNA. You have already studied the mutation in a genetic code in a 5th chapter. That is the point mutation. Point mutation means if a mutation takes place in one base pair, then also some changes will be occur in the formed protein, and because of that particular expression is there, it will be changed. Example is sickle cell anemia. In that glutamine is replaced by valine, and because of that sickle cell anemia disease is formed. This is the example of a point mutation. Now, when this mutations occurs in genetic code, that time some of the triplet codons will also be changed from the point of a mutation. That is called as flame shift mutation. Now we will see how this flame shift mutation occurs. See here, a mutation that adds or delete both, whenever it is added or deleted a base, it shifts the DNA sequence, and this is called as a flame shift mutation. Take an example. This is a normal DNA sequence having CAG, CCC, ACT, which will be code for an amino, the amino acids like CAG will code for glutamine, CCC will code for proline, and ACT for threonine. Now, if any one base pair is added, you can see here T is added here in between G and C. Because of that, the first 3 successive nucleotides are taken as one codon. Then next 3 successive will be taken as a codon 2. So in that T, then next 2. So TCC, codon 2 becomes TCC, and this C is shifted ahead and it will form then CAC, codon 3. Because of this shifting of one codon, here forming amino acids will be changed. Earlier these 3 codons are forming these 3 amino acids. Here first codon is the same as it is, so first amino acid is as it is. Here T is added. So due to addition of base, mutation takes place, and instead of proline, serine amino acid is formed. Codon is there, it will be formed the histidine. So in this way, due to adding one base in this codon, all the sequence of amino acids will be changed. So this change is called as a flame shift mutation. Now, whenever this addition will be takes place in 3 codons, means fully triplet will be added anywhere in this sequence, then there will be no any change. See here, codon 1 codes for glycine, then T triplet is added, so that triplet will code for one particular amino acid, so that amino acid will be added, and rest these 2 codons are there, second and third, it will becomes third and fourth, and their amino acids will be as it is ahead. So here no change will be takes place in amino acids, it will be readable. So whenever triplet will be added, only one amino acid will be increased in the chain, otherwise further complications will not be occur. But when 1 or 2 bases will be added, that time ahead sequence will be changed, and because of that codons will be changed their sequence, and due to change in codon sequence, triplet is changed, amino acid sequence is also changed. So this is called as a flame shift mutation. How it is done for the addition? Like that, deletion also takes place, and because of the deletion also changes takes place. Take this another example of flame shift mutation. A single base pair is added or deleted, then it will shift a reading. Whenever base is added, it is called as insertion, and whenever base is removed, it is called as a deletion. See, this is a DNA sentence, that is the boy cut his lip and ate the hot dog. So these all words are there in the sentence representing the triplets of the codon. So any one of that will be added, any among here any one will be added, then ahead this sentence has a particular meaning. Once any one letter is added in this, the sentence meaning will be changed. So here insertion takes place of S after his S is added here before L. So all these triplets ahead go on changing and becomes, instead of leaf it will become SLI, pan, DAT, ETH, EHO, TDO. So everything is changed here, which is a meaningless. Same thing happens in the DNA whenever any one base pair is added. So this is what it is called as a frame shift mutation due to insertion. Now frame shift mutation due to deletion, same sentence is taken here. The boy cut his lip and ate the hot dog, and here the deleted base is shown here, and A and D is there. So D is deleted. So as D is removed from here, next codons A is shifted here and it becomes A and A. And because of this shifting, one letter shifted ahead to form a triplet, and further all meaning has changed. So this also change in meaning or change in amino acid also takes place due to deletion. So frame shift mutation takes place due to insertion or due to deletion. This is the example. Even in your textbook, one more example is given of Ram has red cap, and in that very well it is explained. That example also you can write whenever it will be asked in an exam. Now next point is tRNA. As you know that RNA is of various type, mRNA, rRNA and tRNA. Earlier we have seen mRNA, messenger RNA. Now this is a tRNA. Actually this tRNA is their clover leaf like model or hair loop like structure. So in this you can see this is the 3' end of the tRNA. Then it comes, forms a stem, then loop, then varial loop is formed. Then here anticodon loop is there, stem is there, anticodon stem. Then again one more stem and loop is formed and comes here at a 5' end. Or you can go from this side also. So this is what a structure of a tRNA. So here this is the 3' end and 5' end. 3' end is there. This is the end where the amino acid will be attached. So that's why this stem of the tRNA is known as the acceptor stem. This loop is called as T loop. This loop is called as D loop, and this is called as anticodon loop. Here triplet of anticodons are there. The function of every loop we will see ahead in detail. First take the D loop or DR. Actually this has 4 base pair stem, 1, 2, 3, 4. So these 4 base pair stem is formed and which contains dihydrouridine. Here it is this D arm is there. This is actually a recognition site for a specific enzyme, amino acyl tRNA synthetase. This enzyme actually helps to form a tRNA combining with the amino acid to form a complex that amino acyl tRNA. So this enzyme synthetase and specific enzymes recognition site is there is of D arm. It plays a very important role in stabilization of the tRNA tertiary structure. This clover leaf like model is there, that is the tertiary structure of the tRNA. So it will be stabilized because of the D loop. The next anticodon loop is there. In this anticodon loop it will be at the lower side. So it also contains stem and loop. In this stem, 5 base pairs are there. 1, 2, 3, 4, 5. So 5 base pairs are there, and there 7 unpaired nucleotides are there. 1, 2, 3, 4, 5, 6, 7. Out of that, these 4 are there this side, and this middle 3 are there. These are called as anticodons. Now these anticodons are there. They are actually has the pairing with the codons on the messenger RNA. So whenever the messenger RNA's codon is read by rRNA, that is ribosomal RNA, the specific tRNA which is having the matching anticodons with this will transfer along with the specific amino acids and get paired on the messenger RNA to give the amino acid. So that's why it plays a very important role in the translation process. Anticodon recognizes and codon of RNA and bind it to leave the amino acid. Next loop is that T loop or tCR. Now in this, if you will see, it is named as tCR. Why it is named? Because it has a sequence of thiamine, pseudouridine, that is Psi, and cysteine. Here you can see thiamine, pseudouridine and cysteine. So because of this sequence, it is named as TYCR. This is also has a stem and loop. So stem contains 5 base pairs, 1, 2, 3, 4, 5. So 5 base pairs are there. Then outer loop is there, that is of CG loop. Then after this 7 unpaired nucleotides are there in the loop, and among that Psi is there or pseudouridine is there. This loop contains a ribosome recognition site. So the function of this is ribosome recognition site. Next, that is 3' and 5' end of the tRNA. Out of that 3' end is there. I said already that 3' end is there where the amino acid will be accepted or amino acid will be attached to the tRNA. So here this is the structure. This is the 3' end where 5' CCA to 3'. This sequence is there. This has OH bond. On this OH bond, attachment of amino acid will be takes place, and this amino acids COOH bond will be joined with OH bond of adenine in the CCA, and amino acyl tRNAs will be takes place. So this is what the structure of the tRNA, where all the loops we have seen. In the exam, based on this, questions will be asked. The diagram of tRNA is very important. So based on the diagram of tRNA, questions will be asked. Functions of their loops will be asked. In a NEET, some of the stems base pairing or their free nucleotides are there. On that number also question will be asked. Their other functions are there, that will also be asked. Another thing is that diagram, drawing a diagram of a tRNA will be asked for 2 marks or 3 marks. Next, mutation, we have studied flame shift mutation. In that also definition of the flame shift mutation will be asked. Ask for explanation. So in the explanation, whatever the textbook example is there, that also you can give, or you can also explain this along with the DNA or RNA sequence. So based on that, deletion flame shift mutation or insertion flame shift mutation you can explain. So overall on the mutation, 2 marks or 3 marks question will be asked. Even on the tRNA, 2 marks, 3 marks or even 1 mark question will be also asked. Among these both, tRNA is very important. So practice the diagram of a tRNA. All the best.